So fairly briefly, at the end of this chapter, they kind of wrap things up by going through the two other types of muscle, cardiac muscle and smooth muscle. Now, cardiac muscle you'll really get into in chapter 20, which is in A&P2 as we start talking about the heart, but we should at least have a basic understanding of some of these differences here. So cardiac muscle, right, is still striated. Okay. So remember, if we looked under the microscope, we could still see striations, which tells us that this muscle is arranged in sarcomeres, similar to skeletal muscle. We did have this difference. We had these intercalated discs that we found in cardiac muscle. And if you'll recall, this intercalated disc was basically where neighboring cells are meeting, and the intercalated disc was basically a lot of protein connecting the cells because we needed gap junctions to let ions basically run from one cell to another. This ends up being super important actually in the heart so that depolarization can actually just spread from cell to cell. In skeletal muscle, you had a really long cell that action potential ran across the sarcolemma. Here the cells are much shorter, right? They're no longer myofibers, they are myocytes. They are muscle cells, they have not fused. And so you need these connections at the intercalated disc. You need the desmosomes, the pores, the gap junctions, the pores to let ions through, and then the desmosomes, this is maybe a better picture, that really anchor those cells together so when the contraction occurs, they don't get pulled apart, right? So that's what we were looking at with those intercalated discs. Other things to keep in mind here, cardiac muscle definitely differs in that it's involuntary. Sorry, if you need intercalated discs, and again, those have the gap junctions and the desmosomes. Cardiac muscle is involuntary. It's also autorhythmic, or some places you'll see it automatic or automaticity. Basically, these cardiac muscle cells can contract on their own. They do not need a neuron to come tell them to contract. Basically, the cell membrane's a little bit leakier. Some of them depolarize a little more rapidly and can trigger the depolarization in the rest of the cells. In the heart, those cells that depolarize the most quickly are called the pacemaker cells. And so they can contract on their own. So that's a little bit different. As you might expect, cardiac muscle is extremely aerobic, right? So more of that slow oxidative kind of fiber. It has a really rich supply of things like myoglobin. It is, of course, extremely vascular. The muscle stores lots of glycogen, has tons of mitochondria, right? All the things that we would expect there, because that heart needs to keep contracting, right? Hundreds of thousands of times. Okay. On to, sorry, one more thing. I thought this was a really interesting picture that they included. Notice how much smaller the SR is, sarcoplasmic reticulum. Where this becomes important, right, if the SR was storing calcium that allowed the muscular contraction to take place, cardiac muscle gets less of its calcium from the SR and more from extracellular calcium. The sarcolemma is actually very permeable to calcium. It rushes in for contractions to take place. And so what that tells you is blood calcium levels, like we were studying how the bone, right, maybe is this reservoir for blood calcium. Cardiac muscle is much more sensitive to changes in blood calcium levels than skeletal muscle is. Okay. to smooth muscle then. Okay, smooth muscle is way different, right? So smooth muscle is not striated. It does not have sarcomeres, right? So the arrangement of actin and myosin is actually going to be quite different. These cells, they're kind of more spindle shaped. They have a single central nucleus. You might even remember that from the tissue chapter. When we look at smooth muscle, typically it's going to be arranged in sheets. So here they're talking about how on a visceral organ, maybe the intestines or something like that, if you think about the intestine as a tube, you may have in one layer cells that are kind of running this way around the tube, a circular layer, and then further out, cells running this way, longitudinally, following the length of an organ. So smooth muscle cells are arranged in sheets like that. Now what that sets you up to do by having cells running in different directions is something called peristalsis, this wave-like motion that smooth muscle can make by kind of alternating which of those is being contracted, and this helps propel food, say down the small intestines, right, or actually propels urine down the ureters, or propels a baby out of the uterus, right? The uterus is also made of smooth muscle. Now, physiologically, I guess I'm sticking with this picture. Physiologically, there are some other differences, right? So we talked about how actin and myosin are arranged a little bit differently. They have what's called a dense body. This is actually going to be kind of what attaches the actin often to the sarcolemma. This is where neighboring cells would attach at these dense bodies. And we end up with this more kind of spiral arrangement. So when the actin and myosin cross-bridge, because that is the same, these cells can kind of corkscrew together. One other difference, these cells don't use troponin for calcium to allow that actin-myosin cross-bridging. They instead use what's called calmodulin. Right, that's instead of troponin. Cardiac muscle still uses troponin. In fact, that's a protein that would actually get released into the bloodstream following a heart attack, right, where there's massive damage of cardiac muscle. You're actually measuring troponin levels in the blood to tell that someone had a heart attack. Okay, another thing to keep in mind here is we use different neurotransmitters to talk to some of this smooth muscle. So, for example, epinephrine is going to, so epi and norepi, epinephrine and norepinephrine. I know we haven't really talked about anything except acetylcholine at this point. This is just another neurotransmitter. The interesting thing with this is depending on the location of the smooth muscle, this can have totally opposite results. So I always think this is kind of interesting. Epi is actually going to constrict arteries, right? It makes them vasoconstrict. It makes them smaller. So if a blood vessel is wide open, right, it's going to bring it in nice and tight. In fact, that is a treatment for anaphylactic shock. In anaphylactic shock, someone's having an allergic reaction and their blood vessels have all dilated and their blood pressure is plummeting. You give them epi to kind of squeeze all those blood vessels in and return blood pressure. It's kind of like the Spanx of blood vessels. But it actually does the opposite. It actually expands the bronchioles, the airways, which is actually quite useful because also in anaphylaxis, often the person's having difficulty breathing, the airways are actually collapsing, and epinephrine is going to help them expand. So kind of interesting that the same neurotransmitter can have different responses depending on where the smooth muscle is. You'll see smooth muscle when we get to the digestive system. There's all sorts of other super interesting things about it. It's actually much more efficient. It uses less energy, even though it can contract just as strongly. But I think we will leave it there for smooth muscle.